Yes. Link should work, yeah. And it should be, if she hits play, it should, on the dark screen should go clean. Streaming now. I'll tell my aunt.
Is that you lagging?
No.
Oh, hello, Andrew. Oh.
Should say, make sure you sit play.
Is Stella going to watch this as well?
We didn't set her up with the thing.
She's going to hit you.
I'm glad you didn't.
She's going to be angry forever.
You have it recorded, it's fine.
You're
You can play it back for her.
That's right.
She'll care like a lot. Yeah.
I'm gonna run to the restroom and then see, make sure the stream is going.
Okay. Are you guys checking out Twitch? Are you guys checking the stream?
My aunt, I think, was checking it, but I'm not recorded.
You're so popular.
Oh.
UWGS one. We can see if.
Hello.
Oh.
You're not going to talk from over there, are you? Are you going to speak from over there?
No.
Okay.
That'd be pretty cool.
Everything you thought you knew about the software.
It's a wireless mic. Can do anything.
Oh, this one.
Okay, nice. Yeah.
Okay. Well, let's see.
Well, we can either sit. I think it would be better because then I think we could see what goes wrong.
Yeah, sure. Wow. Very first time I see you. I think last time I was sitting here when we go live.
I can. Hey, how's it going? No, thanks for coming. Yeah, thanks.
Yeah, so what's the process after this? Is there anything or?
I have to go. Actually, if someone has to print out a thing, actually, which I didn't. Okay. Yeah, I think there's like something that has to get signed, but then and then there's like food and stuff. But if you're not free, it's fine too. Yeah.
So so it's really there's like a oral examination type thing.
I think after there's I think there's supposed to be like questions at the end where everyone else leaves. You guys can ask questions and stuff. Yeah, I don't know what the format is. Yeah, I've never seen it. Okay, cool. Thanks.
Sure.
Hey, Molly. I guess I'm probably just better.
Oh, yeah. 5 years. Go down and have access to it. Forward it to me and then we could print it.
You could just print it, but then I can just grab it.
Yeah, I'll bring it back down for you. How about when you're being quizzed?
Okay, I'll forward it.
Okay.
How do you do?
Jay is like his first service.
She does.
Is there sound? What's that? I was going to present first. Just whatever's done. How's it going, David?
This guy is not.
At the same time.
I realized I forgot to start it.
Oh, maybe I should change it too. I don't think mine.
It should stay awake.
Yeah, I just stay with. Maybe I'll just release in case.
Okay. Since recording.
Oh, can anybody? Wait, Molly, don't go.
You need help?
I just want to say, screensaver is set to never, right?
Start after never.
Okay. And that's it, right? Like,
I think it'll be fine too. I'm sure it's positioned
Okay.
not to do that.
there is no.
You're muted, so you should be good.
Yeah, like there's no. Yeah. Okay. I think that's the only thing, right?
Yep. Sounds good.
There's no like power saving. Hold on.
I wouldn't worry about it.
Okay.
Sorry if the power goes off. You streamers.
Oh yeah, never.
Great.
Yeah. Oh
Oh
wow.
my gosh. Oh my god.
Okay, hello everyone. Welcome to the dissertation defense of Andrew Hill, pictured here, a PhD candidate in the UW Genome Sciences program. Andrew is the 16th graduate student from our lab to defend his thesis, on the 2nd this week. Making this introduction on behalf of both our lab and Cole Trapnell's lab. Andrew's been effectively joined between our groups for the course of his PhD. Okay, so lest anyone think that Andrew is a one-dimensional person, purely focused on single-cell biology to the exclusion of everything else, I'll start with a few words and slides about his pre-genomics background. So unfortunately, Andrew Hill is a very common name, so Googling was not as effective despite my considerable skills. But I know his wife, so that helped. So Andrew grew up in Connecticut. He claims to be of Irish and Scottish descent, but we've never actually confirmed that. Today's our chance to check since his dad's here. As a high school student in Connecticut, he actually went to nationals in gymnastics. And then was on the gymnastics team, men's gymnastics team here at UW. As one might expect, there's proof on the internet. Here's another shot. Here he is. So Andrew was certainly not one-dimensional while here at UW. This is something else I found on the internet, so I'm going to present this with no context at all. So it's been playing for a while. So there's it's just overloading the system. So there's a it's kind of flipping back and forth. Anyways, there's a lot of things I found awesome about this, but so he's either trying to become the $6 million man here, but it's actually he's trying to make a haptic-enabled glove for musical performance, is what it actually is. But I think probably the most stunning thing about this is that Andrew has not aged a day in 7 years. It's a little bit freakish. I was trying to date this picture. I was like, "You're doing this on the side?" And then I was going back and figured out this is from 2012. So good genes. Good dad. Okay, so after graduating, I'm going to stop this. So after graduating from, after graduating from UW with honors in bioengineering in 2012, Andrew pursued his dream of completing this transition to a bionic man, taking a job as an engineer at TechScan in Boston. The paths we take in life, I think, often come down to really chance moments, and I think this is a great illustration of this. In Andrew's case, my understanding is that this took the form of a conversation at a happy hour with a, quote, random husband, as Molly put it, of a coworker of hers who turned out to be Monkol Lek, who was a not-so-random postdoc in Daniel MacArthur's group and was the co-led the EXACT project. And so that led to, I think, a volunteer position in the MacArthur lab, which is something you never know what that actually means in terms of taking it seriously on graduate school applications. This was just a note I got from Daniel MacArthur around the time, who's not one to give him praise, and we had the good sense to accept Andrew to graduate school in genome sciences, despite his pure volunteer experience, and that the rest is kind of history here. So including the fact that he TA'd genetics, despite never having actually taken genetics. Okay, so Andrew ended up rotating in my lab and then Cole's, and then thankfully joining. As I mentioned, he's effectively been co-mentored by us. One thing I think is worth noting, just because it was such an impressive thing, is that he, despite having limited background outside of the glove, and he took the plunge and did well over a year in the wet lab, earning his chops, and together with Jose, doing some really terrific work that you'll hear about today as part of his PhD. So as usual, I'm going to avoid summarizing the work, as you're going to hear about it shortly, but I just want to comment on some of the broader things that I really loved about having Andrew in the lab. First, I think it goes without saying, if you know him or you've spent time around him, that he's absolutely brilliant. I think he's often the smartest person in the room, and certainly when it's our one-on-one meetings, he is. You wouldn't know it because he never, certainly never acts like it. But that brilliance has been the source of many of the critical advances in our labs over the past couple of years. It's also been a source of reassurance, and I think the reassurance aspect is, I think that brilliance is one thing, but in Andrew's case, it's matched to a level of intellectual rigor and care that I'm not sure that I or we had anything to do with, but I'm really proud to have been associated with. It's tempting in genomics, especially when you're surrounded with large amounts of data, to sweep things under the rug or to notice bright shiny p-values and things like that, and to not necessarily be sufficiently careful, as a former student put it, Jacob Kitzman, to really kick the tires, right? And Andrew kicks the tires, kicks them again, keeps kicking, and as a consequence of that, I think the quality of the work, not only in terms of originality, but also in terms of rigor, and then on top of that, transparency and honesty is really sets the bar, right? In a way that I think is even, not that I was dishonest before I met Andrew, but it's a bar that I nonetheless aspire to. So, the right way to say that. But, so, just to go even further than that, just watching how Andrew interacts with others in the lab, his generosity with his time and his talents, but also the problems that he chooses to work on, the manner in which he goes about deciding which problems are worth working on and which ones are not. how he thinks through which places are, when as he was going through his job search, which places are, worthwhile to work for, and which are maybe not, or that you're comfortable in. And all these things, he's had like I don't know, I was going to say a monk-like purity of process, but I had it in there, Phil Green-like purity of process. That is just a really awesome thing. Yeah, so if his, and just one last notice, if his parents, I'm sure this will make an appearance later in the talk, but if his parents are here wondering if they made it into the acknowledgments of his papers and things like that, I want to just know where they stand with Andrew, as he didn't. This cat did. Okay, I'll shut up now. Andrew will have to the end of the hour to present his seminar, and then we'll take some questions from the general audience, after which you'll all excuse. You'll have past 3:30, where there will hopefully be a party if he passes. And his thesis committee will stick around to ask him some hard questions. So without further ado, Andrew, switch this back.
Great. I'm glad I won't be the only one contributing cats to the presentation. Great. No, thanks, Jay, for the introduction, and thanks everyone for coming today. And also thanks for the people who are hopefully watching on Twitch, which is kind of a unique use of Twitch if you're familiar with the platform. We're not playing Mortal Kombat today. Cool. So I'm going to present a subset of my work that all kind of at least tries to address a similar problem, which I think will be helpful in kind of hopefully making it a little bit understandable. So in organisms that you might be familiar with, say a mouse or a human, there are obviously many specialized organ systems that accomplish the many diverse tasks that the human body has to actually, or mouse in this case, has to actually accomplish to make everything work. So obviously it should be fairly intuitive that the brain, for example, does something very different from your lungs. What's maybe a little bit less intuitive if you're not a biologist is that the actual cells or the units that make up these tissues are also themselves incredibly unique and specific in their function. So for example, a neuron in the brain and a B cell in the bone marrow, for example, would actually look very different and be capable of very different tasks in the body. And part of the way they achieve this is that each of these is actually manufacturing a different set of tools or parts to actually accomplish each of these unique tasks. So for example, neurons are involved in neurotransmission, which might, for example, influence your kind of pet preference. Whereas your B cells would be heavily involved in your immune system and would manufacture a whole set of tools that would help defend against your nephew or try and thwart your cat, like affinity for cats. So each of these is actually making use of a totally different set of tools, despite the fact that they all, fairly interestingly, all share exactly the same DNA or the same genome. So this tremendous cellular diversity is not achieved through sequence differences in your DNA. So how do we actually generate this diversity given the same exact underlying set of instructions? So one way that you could probably intuitively think this might happen is that there's some mechanism for a cell to either make a protein or not make a protein, to kind of selectively combine these tools into useful kind of functional units like a cell. So the way that this will be a very rough kind of schematized version of what actually happens, but in a cell you could think of it as there's segments of your DNA that contain the instructions for making a protein, or the machines that actually go and do things inside your cells. We typically refer to that segment of DNA as a gene. And without going into details of why, these genes in order to become a protein, these genes first need to be made into RNA, which is kind of an intermediary molecule, that then basically tells the cell, I want to make copies of this protein. This RNA, then there's machinery inside the cell that would convert these RNAs into proteins, which will actually go and do things. So you can imagine that one mechanism for deciding whether or not to make a protein is to just decide whether or not to make RNA, although there are certainly many other mechanisms by which protein levels are regulated. So the way there are two kind of types of switches that I'll talk about today that allow a cell to kind of turn the expression or the generation of RNA from genes on and off. One type of switch is a promoter, which is basically the sequence immediately upstream of the gene. And so these are capable of recruiting all the necessary components to actually turn this gene into RNA. And then there's another type of sequence that I'll call an enhancer. It's basically, you can think of it as kind of serving a similar function to a promoter, but it's actually located much farther away from the gene. So they're actually much more difficult to study because there's not necessarily a easy, intuitive way to associate a given enhancer with the gene it's regulating. So the whole kind of talk today, I'll talk about two different main ways that you could hope to study kind of promoters, but probably more interestingly enhancers in this case, and how they function. Yep.
Is the relation between enhancers and genes one-to-one?
No, I think there's lots of examples of both enhancers that regulate more than one gene, and also genes that are obviously regulated by more than one enhancer. Yeah, exactly. And so one of the ways, for now, I'll just assume that we have some experiment or some protocol that we could do that would kind of very roughly tell us whether an enhancer is likely on or off in a cell, which would kind of give you some indication of how the gene is being regulated in the cell. If you did that, there are a couple of things you could do to study enhancers. One of them is you could actually just measure the state, the on or off state of enhancers across many different cells and many different tissues across an organism, kind of examine the genome in its natural state and measure whether enhancers are on and off as a way of cataloging and then later leveraging that data to understand more about enhancers. Another way that's maybe a little bit less intuitive is that you could actually just try and take a homogeneous population of cells and break enhancers and then see what happens. So this would be kind of perturbation experiments where basically you're just trying to establish a more causal relationship by breaking something and then seeing what happens in that broken state. So both of these present several technical challenges, and I'll talk about both of them, but I want to start by talking about some of the challenges of actually trying to make these enhancer on or off measurements, even just in this first case, in this mouse case. So hopefully you could agree that a kind of ridiculous experiment that you could do would be to take all the cells from an entire mouse and to lyse them and then perform your experiment on all the cells from that mouse. That probably wouldn't tell you anything specific about any one tissue because you'd be measuring the average of what's an incredible collection of different cell types. So maybe less intuitively, also kind of ridiculous experiment would be taking an entire organ, say the kidney, which also happens to be composed of many different cell types, even though they all kind of collectively serve one function. So the nephron in the kidney is just one structure within the kidney that actually has many different diverse cell types that allow it to collectively carry out its function. So if we perform an experiment on this entire organ, what we would get is just an average profile again, where these kind of peaks in gray are supposed to represent kind of like signal from your experiment telling you whether an enhancer is on or off over the genome, which is kind of the black bar. And I'll kind of use this visual motif throughout portions of the rest of the talk. So what we'd actually like to do, in an experiment that's probably less ridiculous, is to actually have some way of obtaining individual experimental results from the individual cell types that actually exist within the kidney and are actually kind of carrying out their individual functions. And today I'll talk about one way of doing that, which is using a set of technologies called single cell methods. So here I'm going to show, just because I think it helps with the intuition for how you actually would do an experiment like this. I'm going to show over the past decade roughly some of the major landmarks in terms of publications as to how single cell technologies have evolved over time. And on the y-axis, I'm showing kind of a rough proxy for the scalability of each method, that is the number of cells that you can actually collect data on. So probably the most intuitive but least effective way that you could do this experiment is to take one way to get a homogeneous population of cells such that you're not averaging in a harmful way. So literally take one single cell and put it in a tube and then do your experiment on it. And that if you did that many, many times, you would have collected data sets on individual cell types. However, that obviously doesn't get you very far because you can only do one cell at a time. You can also multiplex these in wells, so plates with wells in them, and then you could do hundreds at a time. But you could imagine that if you're just kind of taking cells from any given organ, this still wouldn't be of sufficient scale to really tackle these kinds of problems. So fortunately, I entered graduate school right in 2014, just kind of at the tail end of a wave of technology development that led to first, basically automation or partial automation by robotics or microfluidic devices of these well plate-based protocols to kind of, if you had a lot of money and a lot of other resources, you could get data sets that are kind of in the few thousands of cells kind of size, which is starting to approach the scale of the problem. The unfortunate thing is that these methods really weren't accessible to most labs at the time due to the sheer cost of doing these kinds of experiments. So there are a couple ways of additional technology development that have really helped kind of basically bring these methods into the kind of more mainstream. One of them is droplet-based devices, which are also now commercialized. And you can kind of think of these as if, say you were underwater and you blew bubbles of air out into the water. They don't just disappear. You kind of get these contiguous kind of air pockets within the water. So these devices, very roughly speaking, are basically blowing water bubbles into oil so that you can have a little miniature reaction volume inside oil, kind of partitioned inside oil. And what you can also do is you can also, as you're blowing that bubble, incorporate cells and other reagents you need to do a reaction into those same bubbles so that you can do single cell experiments without ever physically isolating all cells in a plate. So these have been tremendously more scalable than the previous methods, and there's been many progressively larger and larger experiments that have been reported with these devices. And we've been fortunate enough that we've also seen the development of other methods in our group and others, such as combinatorial indexing, which I won't talk about in detail today. But these are for, basically we now have several sets of technologies that have different trade-offs with respect to cost, throughput, and flexibility, and data quality. So now we can collect single cell kind of data from lots of different cells because of these technologies. So now we want to do something useful using these technologies. So the first project I'll talk about today is what I alluded to earlier, which is this kind of collection of a different technology, single cell ataxy, which I'll talk about what that is in a second, over many different tissues in mice. And then I'll talk about how we can kind of take a different view than doing perturbation experiments, but also leveraging these single cell technologies as a way of reading out these assays. So the bulk of this work is actually described in this paper just last year. And I want to say up front that this was a very large collaborative project in which I was fortunate enough to play a major role. But this was during Kuzanovic and Riza Daza had already developed the method that I'll describe in some detail. And then they had also already collected all the data that I'll show you today. I've been fortunate enough to kind of continue to work with them, as well as another postdoc in the lab, Sylvia Dumka, on extensions of this work since then. And this is also a collaboration with Hannah and Delasa and Cole Trentwell's lab, as well as many others, like the Distish lab. So the basic premise of the assay that we're going to use here, so this is the experiment that I alluded to earlier that can tell you, roughly speaking, whether an enhancer is on or off. And the basic principle that it uses is actually much of your genome is physically compacted very tightly just in order to fit the genome inside the cell. So much of your DNA is actually very tightly wrapped around these things called histones, and physically inaccessible to any proteins that might want to get at it. And only the things that actually need to be doing things and be accessible to cellular machinery are actually physically unwrapped enough and open enough to be accessible. So these might be things like enhancers or promoters, obviously, since they need to be able to interact with the kind of machinery that will allow the gene to be turned into RNA. So if we had some way to basically selectively measure which portions of DNA are accessible, we'd have at least a reasonable proxy of which regions of the genome might be putative enhancers. So one way to do this is ATAC-seq or ATAC-seq, depending on who you ask. And what it does is it has this, is basically a thing called a transposase, which is basically, you can think of it as just, you give it some DNA sequences, and it will preferentially insert those DNA sequences into regions of open chromatin, simply because it can also only physically access open chromatin. So since we know the DNA sequences that we actually put on this protein, we can actually then selectively amplify the regions that were that actually had this transposase integrate DNA into it. So you can get signal preference. We can then figure out which parts of the genome actually were accessible by telling where these transposases actually inserted into the genome. So Darren and Riza collected data from 13 different tissues across mouse, several in replicate, and collected kind of in total about 100,000 cells, which by today's standards is fairly small, but it represents kind of a shallow pass over each of these organs. And so there are many goals that many things that we did with this data set, which I won't be talking about today. I'm really just going to talk about, because I think it's maybe a little bit more understandable, is how you would actually try and identify cell types in this data set when really all you're measuring is chromatin accessibility, which is kind of this abstract concept that's not linked to most of the past literature. So when you get this data, unlike if you just collected data on a lung or muscle or something where you have the sample in hand, you know what it is, you've dissociated cells from an entire organ and actually don't know a priori what each cell in this big data set that you collected actually is. So there are two main goals once you get a data set like this that you have to do before you can do anything interesting. And these are not trivial in and of themselves, but I'll just kind of broadly go over what they are. So the first one is if each of these dots with the same color, or each of these dots is a cell, we first need to kind of group cells by which one, by kind of their cell type, or which group cells by similarity to one another. So for example, you might have noticed that there are 5 different groups of cells, all of which have the same color. So our first goal would just be to kind of simply group them into functional units that seem to be kind of hopefully cell types, but could represent other kind of groupings of similar cells. So now your task is to actually tell what cell type actually is present in this data. So one way that you might do, so this is kind of a challenging process, but there are a few different ways that you can break this down. One is that you can go back to the literature, and there's lots of literature based largely on immunohistochemistry, so protein level markers and RNA level markers, where we know that certain cell types, say in a complex structure like the nephron and the kidney, we know that there are certain proteins or RNAs that are very abundant in certain cell types. So we know that if we look in our data set for those same markers that were identified kind of via other mechanisms, we should be able to hopefully identify the cell types that we would expect to see in the kidney, but in our data set. But now the advantage of our data set is we'd have this entire new kind of enhancer assay having been run on a cell type where we never would have had that data before. So just like the plot, a little bit more complicated than the plot I showed you earlier, this is similar in that each dot is a cell, and I've kind of grouped the cells such that cells that are more similar to one another tend to be closer in this plot. And you can computationally group cells kind of into similar, basically distinct groups. But now our goal is to actually identify what cell types we've seen. So one approach would be, I'll just focus on this one cluster here, and I'll tell you ahead of time that these are endothelial cells, which amongst other cell types are kind of make up collectively or circulate kind of the kind of circulatory system. So so basically we went to the literature and there's a gene, say FGD5, which is the name of a gene, or something that will eventually become a protein, and it should be orders of magnitude more highly abundant in this cluster than anything else. And if we were measuring RNA, that would probably be true. But what we're doing is measuring this kind of accessibility quantity, which is much more abstract. So often what you'll see is that if you just plot kind of the signal that you get over the promoter, that is one of the switches that can turn this gene on or off, you'll often see something fairly dispersed like this, where you'd be probably hard pressed. So if each dot in black is something that has signal at the promoter, you'd probably be pretty hard, it'd be hard to convince yourself that the thing I have circled is the only thing expressing this gene just from this data set. It's kind of confusing. So why might that be the case? And just to verify the kind of visual thing, if you actually do this quantitatively and take like a normalized average of this promoter signal, the right thing is at the top, but it's not very well separated. So how can we fix that? So what's probably going on is that at the promoter of this gene, if you look at the signal from our assay in our cell type in orange and then some other random cell type in the data set, what's probably happening is that you actually have some accessibility, even though there may be slightly more accessibility in your correct cell type, there's really not a lot of difference between the two. And remember, I told you that there are two kinds of switches that can turn a gene on or off. And often in these cases, which don't make up all cases, but they're fairly common, you'll see other sites, probably enhancers, away from this promoter that actually are kind of on in this cell type and not on in others. So this is probably what's accounting for the kind of differences in actual protein or RNA abundance, but it's not something that we would have seen in our data set without incorporating these enhancers. The tricky part is that there exists basically no good map of which enhancers actually regulate each gene. It's actually a trickier problem than you might imagine. So actually trying to incorporate this in some way, some meaningful way, is kind of tricky, but it turns out that you can kind of do a few kind of fairly rough approximations, and it tends to work quite well, or well enough. So this is where we started, which is just looking at signal over the promoter. And then again, this didn't work all that well. A kind of naive thing that we could do is take each enhancer around the area and assign it to its closest gene, and then just add up all the counts from all of those switches that could be turning this on. And in that case, the cell type at the top, which is the one we want to be most highly expressed for this gene, is a little bit more distinguished from everything else, but still not great. And we've also done something a little bit fancier where we used a program called Cicero from Cole Trapnell's lab, made by Hannah Pleiner, which basically the upshot is that what we're trying to do is weight these other switches more highly if they're more strongly correlated with the signal at the promoter. So if they kind of look enough like the promoter, we'll weight them preferentially over sites that don't kind of behave in the same way as the promoter. So this is just exploiting those small changes of the promoter to kind of draw these correlations. And in this particular case, this dramatically increases the specificity, although I'll admit that this represents kind of the minority of cases. We do see like a global improvement, but it's still fairly modest, and we're by no means like measuring gene expression from ATAC-seq data. But hopefully this is kind of good enough and cleans up this enough that you could actually look at markers effectively. But around the time we were finishing this up, it seemed like doing this from ATAC-seq data manually was kind of a silly proposition, in that like it represents an enormous amount of additional work to actually go and re-annotate things that might have actually just been measured by single-cell RNA sequencing, which is measuring RNA levels and is much more easy to correctly interpret. So we thought, right around when we were finishing this paper up, a number of papers actually were published on data from similar organs and similar mice, but using single-cell RNA sequencing instead of single-cell ATAC sequencing. So we wondered, could we leverage those data sets to actually automate this process that was very painful and manual for us? So just to make this a little bit more concrete, the task is some other lab somewhere else, or perhaps someone in our lab, has collected a data set that measures RNA levels, and they've already gone to the trouble of annotating cell types in their data set the way it describes, but it's much more easy for them. and then we collect a new ataxic data set, could we actually just automatically transfer these labels between the two to make our job much easier? So using the methods I told you about to get, where we're trying to arrive at a gene level score of, to try and get something like expression, we can do that for all genes in the genome, and then we've effectively made the ataxic data look at least like a really bad version of RNA-seq data. So, but hopefully similar cell types in both data sets bear enough similarity to one another broadly across many genes to actually pick them out in comparison to other cell types. So without going into any of the details here, what we do is basically we have a method, and there's been other extensions of this since, that basically take both data sets, and then what you try to do is for any given cell in our data set, we look across to the other data set to find which other cells are most similar in the other data set. And we can kind of do a majority classification across data sets to say, well, all of my neighbors in the other data set are an orange cell, so I'm probably an orange cell as well, but in ataxic space. You can march through and do this for all ataxic cells. And so at the end of this, I won't go into the results because it's a little bit tricky to walk through the entire graph, but I'll just show you the one example that we already looked at, the endothelial example. So you can run this, you can do this for all cell types in this example. I'll just focus on this one row and column. So on this axis, going horizontally, this is our endothelial cluster that we annotated in our data set. And the color in the graph, if it's more yellow, it means that more cells were assigned to a given category in the RNA data set. And the only endothelial cell category in this RNA data set that we're comparing to is also the one that we're assigning all of these cell types to, again, automatically in this way. And that there are many other problems that come up in an analysis like this, and this doesn't work for every tissue, but this is just kind of an example that something like this can work and actually does work quite well in general for the kidney. So once you actually annotate cell types, which is arguably like the least interesting end goal of the whole project, you can go and do a bunch of other interesting things, which we've done. And we've actually made all the primary data and the secondary data available online. And I've gone to a lot of effort to actually make this quite well documented. So both the primary data and all the secondary results are very well documented, and both in kind of what they are and also the file formats and everything. It's been nice to see actually several other labs make use of this data set, which is not true of much of our past data and using this technology. So I think that's actually been a really cool outcome of this project. So for the second part of the talk, I'm going to shift gears and talk a little bit more about how we could move away from just measuring things in their natural state and trying to actually modify the state of a cell and see what happens as a mechanism for understanding enhancer. So to explain this, I'm going to actually explain it. I'm going to step back from the problem of enhancers specifically and actually just talk about how a concept called genetic screens, which is one tool for doing perturbation experiments like this, generally tend to work. And this will broadly cover a paper from last year, and I'll very, very briefly kind of hint at something that Molly will talk a lot more about in a couple weeks. So these types of experiments, as I mentioned, start with, rather than many different cell types, start with a homogeneous, basically identical set of cells in a dish. So all of these, if you're looking at an enhancer or a gene, they should all roughly kind of express the same things. They should have the same enhancers on and off, more or less. But what we can do is we can introduce changes into the genomes of those cells. So this could be using CRISPR, or this could be using any of a number of other technologies. Basically change the genome in some way for these cells, such that many different cells in the population will have different DNA changes that we can all measure in a pool. And so this is a very simple example of how you could actually then leverage this diverse population that you've just created via introducing mutations. Let's say that some of the cells, when you break some genes in the genome, actually now gain drug resistance, for example. So only the cells in red would actually be resistant to this drug, to some particular drug. if you then treat with that drug, one nice property of this is that all the cells in white that you don't really care about because they don't have the genetic changes that actually resulted in drug resistance would die. And you could actually very strongly select for the cells that actually now have drug resistance. And then the useful part at the end is that these methods are designed such that you can very easily go back and tell what specific mutations actually gave rise to that drug resistance phenotype. This is a nice way of, in high throughput, mapping genotype to phenotype. So one of the downsides, there are many pros of these assays, and they're used widely in our department, and they're often the right tool for the problem, but there are a few contexts in which we thought that methods like this could be improved upon for certain applications. So one problem with these methods is that they're often not that as easy as drug resistance, so they're often very difficult to design assays that do this effectively. And part of the reason is that the assays are often targeted towards a very specific protein or a very specific function in the cell, which means that they don't easily generalize to other systems if you want to go and study a different gene or something like that. Although there's some nice work out there for more generalizable assays in certain contexts. And another con, which is often not actually a con, but in the context that I'll be talking about as a con, is that they don't directly measure a molecular phenotype. That is, you're not gaining specific information about an enhancer turning on or off, or an RNA being expressed or not expressed. You're kind of you're measuring a higher level cell level phenotype often, or a pathway level phenotype that doesn't tell you about those specific things. Often it's very useful because it kind of takes this difficult to interpret and noisy changes in molecular phenotype and integrates them into a larger phenotypic change. But in some cases, you might actually be really interested to tell if one specific RNA is actually going up or down. Say if you're trying to understand whether an enhancer being broken actually impacted that expression level. But the big advantage of these methods is that they're incredibly high throughput. You can measure the impact of thousands or tens of thousands of mutations per experiment. So what we set out to do is try and measure molecular phenotypes, which I said kind of would be helpful for our application, but also while maintaining some of the throughput of these particular methods. So hopefully, as I've already explained, one assay that would let us measure expression or kind of the abundance of RNAs in the cell would be single cell RNA sequencing. So if we wanted to actually be able to make all these different mutations in the cells, but still understand which RNA changes were made by certain genotypes in the cell, all we would need to do is for each cell actually get the genotype that we genotype change information as well for that cell. So you need to couple these two pieces of information, which is the main focus of the next part of the talk. So specifically, we'll be dealing with the CRISPR screen, and you may have heard of CRISPR. There's very few parts of it that you need to understand to understand this fairly arcane kind of thing that I can present. But one of the important things is that CRISPR depends on a guide RNA. So there's an RNA that you can make the cell express. So often the cell already expresses Cas9 because you've engineered it that way, which is the protein that'll actually go and make the change. But you need to tell the protein where to go. So you need to express this guide RNA that actually has a sequence somewhere in the genome that you want it to go. And so you can we basically just need to add this extra piece of DNA into our cells to actually let it express this kind of RNA that will navigate it to the right place. Unfortunately, single-cell RNA sequencing methods, at least at the time, couldn't directly detect these guide RNAs. So you can't actually just read them off and tell where you've targeted in the cell. You need some other mechanism to actually tell you where you've targeted in a given cell. So fortunately for us, there's already another gene often on this piece of DNA that we're putting in that allows us basically to cells that get this piece of DNA to survive treatment with a drug. And this is useful because we can kill off cells that didn't actually get our construct, which is useful for a number of reasons. And so this gene is expressed. So what we did was we actually put a barcode sequence in the tail end of this gene such that we knew that DNA sequence that we inserted there actually made a one-to-one mapping with the guide RNA sequence. So while we can't detect the guide RNA sequence, what we can do is make the cell, again, express this gene. And importantly, the series of A's at the end of this gene actually mean that we can read this off in single-cell RNA sequencing and actually tell its genotype just by sequencing this kind of proxy. So you may have heard of methods like this in the literature, and it turned out that when we were trying this, we actually saw a marked difference in the quality of the data or the degree of noise. When we did the mutations separately, it was all great, but when we pulled them all together, the signal was suddenly much, much worse than before. We were kind of scratching our heads for a long time as to why this was the case. It was only through a lot of digging and also help from other folks in the department, like Kenny McTrack and Jesse Bloom, that we were able to kind of pin down why exactly this was happening. And to explain this mechanism, it might be a little bit, I don't know how intuitive I'll be able to make this part, but you can, yeah, I'll try, and if you don't understand, it doesn't matter. I only spent like a year and a half. So to get this piece of DNA into the cells that we care about, we actually make use of an engineered version of HIV, which you've probably heard of, but it's engineered to be safe, and you shouldn't be worried about that. And it basically will insert, because the way HIV works is it inserts its own genome into cells, we can get it to insert a different genome of our choosing into cells instead. But to make those viruses, what we have to do is you have to put a bunch of these pieces of DNA, all of which have different guides and different barcodes, into cells to make them express the viral genome and also express the other components that the virus needs to like actually go and infect other cells later. So when you do this, hundreds of thousands of different of these DNA sequences actually go into each cell and are kind of all transcribed together as RNAs. And it turns out that this particular kind of virus is actually packaged initially as a diploid, despite the fact that it only will integrate one viable copy of its genome. And because we've mashed up all these different guide sequences together, it means that any one virus will actually basically almost always contain two different sequences and not be homozygous, not have them be identical. It turned out the machinery inside this virus uses to convert its genome, which is currently an RNA, into DNA that you can put inside the cell's genome. It has a very high propensity for basically, as it's chugging along, say it turned part of the transcript or part of the sequence into DNA, it will then actually hop to an identical region on a different co-package molecule at a very high rate, which is kind of interesting. But then it'll kind of continue on converting to DNA throughout the rest of the transcript. And so the problem, as I've drawn here, is that the product of that would be actually a molecule where blue no longer goes with blue, blue goes with green in this case. So since we're only ever going to actually read out green in our assay, there would be a high rate of basically mismatched guides and barcodes. So we've actually measured, I'm not going to tell you about the experiments, but me and others have designed experiments to measure this in this particular context. And it turned out that the rate is about 30 to 50% of the assignments that you would normally make in this experiment are totally wrong. So that kind of explains why we had the problem to begin with. But if you're familiar with this literature, luckily we kind of thought about ways that we might be able to solve this. So I'm kind of redrawing the same vector. And you can imagine that rather than putting this barcode in this kind of gene that's going to let us kill off cells that didn't get this piece of DNA, we could do something a little bit unconventional, which is to actually just put the actual components that express the guide RNA itself inside this region rather than just a barcode. The idea is that you would have one transcript or one RNA that actually contains your guide RNA sequence directly, and that would be detectable in single-cell RNA sequencing. And then a second transcript is expressed by a different promoter, which would actually go and tell Cas9 where to go. So unfortunately, this doesn't work at all if you do it like I did it. The guide basically doesn't get expressed, which is maybe you'd have intuitively thought. We thought it was worth a shot. But fortunately for us, another paper very quickly put us out of our misery and said that it came up with almost exactly the same design, but if you put this in a very specific portion of the virus, a second copy of it will actually be integrated at a different site, and you'll rescue expression of this gene, which may not make much sense with birds, but I can talk about it more later if you're interested. The design is called CropSeq, which is from Dottlinger et al., 2017. And so with this tool in hand, we could actually go and do the experiments that we wanted to do. But this represented kind of a long haul of a project and to get going. So notably, if you're familiar with this literature, we were actually scooped like 3 times on one day and then 2 more times over the next several months on this project, all using the method that we actually stopped pursuing because it didn't work very well. And the reason that it worked in their case is they actually collected very large data sets or collected them all in individual pools where you never would have had this swapping, and so you couldn't have fixed the problem. So this next paper came out, and we actually wrote up a whole piece kind of describing our experience with these methods and also kind of recommended that people use this last one, which is kind of a nice outcome after having been like scooped a million times over. What was also very reassuring was that after we kind of published that work, several other preprints and papers had come out kind of also describing similar experiences with this problem and kind of corroborating all of the findings we had, but coming at it from different angles. So it's kind of like a new grad student who had just learned MATLAB. It's actually pretty reassuring that lots of other smart people across the world actually didn't really appreciate this problem like either. It's kind of nice. And the part I want to talk about is what we've done since is we've actually used this tool to try and answer the following question, which is, can you tell using a screen like this, at least in some cases, if you break an enhancer, what gene is it actually going to change the expression of, as a way of kind of building at least some examples of connections like this. And so of course you can actually break enhancers in a few different ways using CRISPR-Cas9. And so the work that Molly will talk about in a couple of weeks will focus kind of on our results using this kind of method, but to test this question. So today I've told you about 2 ways to measure, to try and understand enhancers more effectively. One, by measuring their state in many different cells across the body, and then also by breaking them and trying to tell what happened afterwards through perturbations linked to single cell methods. So one thing that I really don't like about dissertations is that they kind of come across often as like a highlight reel of all the things that worked for you. That certainly isn't representative of my graduate school experience at all. And so I've used the term work happiness, which is probably not correct, but is kind of for lack of a better term, over graduate school. And I wanted to kind of highlight a few key points along this graph. So I had some good rotations, and my first few months in Jay's lab went great, and things were still working. This is like right where some of that lentivirus stuff kind of came up, and basically what we've been working on stopped working and continued not to work for a very long time. And also I had an entirely different project that I worked on with Greg Findlay in the lab that completely fizzled out on 2 major iterations. And so that was all happening then too. So I had actually already planned to do an internship at a company called 10x Genomics. And this summer I was really wondering, like, should I even go and do this? Because everything's really not going so well right now. But fortunately I didn't make a short-sighted decision and still did it. So this little uptick is like my time at 10x. And actually to the point where Bob, our former chair, actually when I got back, he actually said like, oh, you came back. When I was locking my bike up for the first time. This is when we got scoops like 5 times over, so things were great, and then I came back and then they didn't go so well again. And then eventually we kind of switched to this more technical direction where we kind of wrote up this paper about our experience in this, and then I also, Riza and Darren were kind enough to kind of bring me on to some of the single cell ATAC-seq works, which has since kind of been a very productive area of research for me personally and a great experience for me overall. And then kind of right now, so lots of things of good things, probably most of the good things you're actually aware of that I've worked on happened in the last like 1 to 2 years.
Looks like you're happiest in your rotation.
So this like delta here, I think, is the result of having gone through graduate school. Yeah. And I just want to note that any uptick in this graph is the product of two things. One thing, and less importantly, was that I made kind of distinct changes in what I was doing. I was willing to change direction when I needed to, or be persistent when I needed to. But I think that by far the most important factor was having colleagues and advisors who were like actually open to allowing me to make those decisions myself and allow me to do independently what was best for me, and also like encourage me and be amazing people around me. And I think without that, any persistence wouldn't have mattered at all. So I want to thank specifically like all the folks in the Shinduri and Trentmore labs, and also specifically Darren Kuzanovic and Riza Daza for all their help on the single cell attack seq work, as well as Silvio more recently. And then Molly Gasparini, who I'll talk a little bit more about later. And then as well as Jose McFallon, who got to share that experience with the whole perturbation work that we did, and we've since been involved in a few other projects together. He's been a great friend. And then Cole and Jay, obviously for being kind of having unflagging optimism in the face of a ton of adversity, and also producing an environment where there's so many awesome people around that kind of it's still fun to come to work today, even when you're on this kind of like continual downslope of things not working. So I just want to thank a few more specific people. So this is a picture of, so the guy in the red is Martin Kircher, and then flooding next to him is Matthew Snyder. And they were actually the first people I worked in together. So Jay alluded to like rigor and methodology and kind of good science practice, and I think getting to work with Matthew and Martin, who are two very extremely experienced scientists, getting to work with them literally as the first people in the department who I ever did a project with, I think was like an incredible opportunity, and like, I don't know that anything else I could have learned in graduate school would have been as useful in the long term as kind of the basic scientific rigor that they kind of helped instill and further instill in me, and probably will last me a lot longer than any specific domain expertise that I could have acquired otherwise. So I'm extremely thankful to them. I'm thankful for a lot of people in the Shinduri and Trentmore labs for being so collegial and willing to have conversations. Darren Kuzanovic, who's in this picture, actually stood out to me as being one of the more kind of on the upper end of that spectrum, and we also talked, had tons of science conversations that I really enjoyed. When I looked back at pictures of Darren, I realized that through like actually a shockingly large part of our interaction together, he was actually in a cast because he had fractured his knee. So I actually am not sure if his presence on like the upper tail of that is due to his physical immobilization or because he actually enjoyed talking to me. But we still talk on the phone, and even though he did move away to Arizona. I've had tons of great friends over the years that have made, even when things weren't going well in lab, tons of fun with everyone outside of lab. And so very thankful to everyone else, all the people that I've met at UW. I want to thank my family, which notably my mom and dad, and my mom, I think, hopefully is watching online from home. And I wanted to highlight two things that I found that were kind of fun going through my own family photos. One was the prevalence of 90s era clothing. And then the second one is the very long lineage of cats that are running through the middle of this collage that have strongly influenced my adult life as well as business. And obviously Molly, so if you don't know, my wife Molly is a scientist. She's a graduate student in this department. She's also a student in Jay's lab, and she actually sits in the next bay over. And we've had this incredible run of adventures throughout graduate school, and I really have no idea how anyone else does it without someone like Molly to like actually kind of make kind of some of the more insufferable parts of graduate school sufferable. Yeah, so Molly's, and she's also literally supported me in that we're actually co-authors on 2 different papers, which is kind of a unique experience that neither of us ever expected would ever happen. Through Molly, I've gotten to meet all of Molly's family, and I included this mostly to show that if you can swing it, like actually, if you need a boost of confidence during graduate school, if you can somehow manage to make your 2-year-old nephew like you way more than their aunt, then that can also be pretty helpful. And then lastly, partway through graduate school, we also got I got a cat named Stella, who, in addition to being a great grad school companion, is an excellent typist, which is why I included, so Jay already showed this picture where we included her in acknowledgements. What I think he didn't show you is that the copy editor from this, at this journal actually inserted a comment asking for cat photos. And so I don't think she had any idea who she was messaging. I gladly sent her many cat photos. And she actually replied to my email, which was super sweet. And so what I haven't told you is that I actually sent her the last thing in my talk, actually. What I did was I also sent her a video. And one of the kind of parallel developments outside of genomics in the field of computer vision has been the improvement of facial recognition algorithms. And through the sheer amount of training data that we've provided Google Photos with of our cat, we can actually type Stella into Google Photos and it'll populate hundreds of photos of our cat. But it also means that we get these great auto-generated videos, one of which I think is actually encapsulated in my graduate school experience far better than anything else I could have possibly told you, at least from a realistic standpoint. So I just want to close by sharing the video I shared with Carolyn.
So RNA-seq is not my field at all, but my understanding is that your data sets can sometimes just be inherently noisy. So when you were taking your when you were taking the RNA-seq data from other labs and trying to use that to do your cell assignments and make that all automated, how much did that play a role or a problem? And were certain cell types just that wasn't an issue at all, and other cell types was a huge issue? How did that work out?
Yeah, I think part of this is a few different problems. Probably the biggest one is that the interpretation of a lot of these data sets is subject to a lot of manual work and subjectivity. So the things that someone is looking for in their data set may not match up in terms of terminology with what we had used, for example. So any labels that you're lifting over would inherently be kind of contaminated by those kinds of errors, so manual annotation kind of errors. And the data sets are noisy, but part of why we use the approach that we did is that it's kind of based on a batch correction approach that tries to basically combine RNA-seq data sets that maybe were collected on different technologies or were sequenced to variable depths or things like that, where there's a large batch effect. And so you're kind of trying to find common signals in the data set that let you kind of bring together the kind of common cell types. And granted, this is a pretty far stretch of that sort of approach, given that we're coming up with RNA-seq data from ATAC-seq data. So that's, I think, in general, I think comparing RNA-seq data sets is much easier, but it also happens to work at least well enough, at least for these high-level, very different cell types to work for our case. But presumably for more difficult to distinguish cell types, this kind of approach would be much more subject to things like noise. Yeah, given the biological signal is lower. Yeah. Yep.
So I remember you modified the Cropsic plasmid and the another version which is OptiCrop. So what was the exact modification with the Cropsic plasmid?
That one was actually, so it wasn't relevant to the initial paper, but it was just modified to have a backbone that was compatible with CRISPR-I, or is basically like a more stable flavor of that backbone. So that it's, and the reason we called it Opti was because that's actually what the original paper that developed that backbone called it. It was an Opti backbone. But it actually just has a longer hairpin section of the RNA that makes it more stable. Yeah, or increased, yeah, it makes it more performant in CRISPR-I. Yeah. Yep.
Can you speculate on why the gene that was inserting the guide RNA to the end of the lentiviral gene wasn't getting expressed? Was it that it wasn't getting expressed at all, or just not at sufficient levels?
It was definitely getting expressed to some extent, but like both editing rates were lower for sure by quite a lot, probably a factor of like 3 or something. And then CRISPR-I really didn't work very well at all. It's like barely worked for the promoter that we were testing it against. I think probably the main reason is that there's not many endogenous examples of overlapping Pol2 and Pol3 transcription, and we think that Pol2, particularly since we're selecting on the Pol2 transcripts, that one's probably fine. But then once you Pol3 probably just isn't. Because those transcripts are overlapping, you're probably just not getting sufficient expression from the Pol3 promoter, which is part of the reason, yeah. But when you actually get a second copy that integrates not overlapping Pol2 at all, it kind of fixes itself, yeah. Yeah. Yep.
You were able to look at a large amount of single cell ATAC-seq data and RNA-seq data. Did you see a difference in how easy it was to cluster cell groups in using the ATAC-seq data compared to the RNA-seq data?
I think it's a little bit tricky to say. Certainly for the data that we collected, I think it's much easier to both cluster and identify cell types in the RNA-seq data. We've since started to look at a few, I think that it's still not entirely clear to me how much of that is because of data quality differences between the two versus actually the inherent kind of ability to use those measurements for those tasks. There have been a few recent papers that are from very complex data sets from droplet kind of based single cell ATAC-seq methods, where actually at least for the cell types that they've been looking at, you actually see a lot of the same story in both RNA-seq and ATAC-seq data. So it kind of maybe partly that, and also partly differences in the computational methods that we're using in both, which are kind of still being developed more so for ATAC-seq data than RNA-seq data. Yeah, that's a great question.
All right, if there's no more questions, let's thank Andrew again. And if you can, please hang out in your S330. So you can always see.